Hi, I'm Svi Pollack here at the University of Pennsylvania and Institute for RNA Innovation. I'm here on behalf of the Vaccine Makers Project to show you guys a tour of what the RNA innovators are working on.
My name is Ted Kreider. I am an instructor in the Division of Infectious Diseases in the Department of Medicine and a faculty member within the Institute for RNA Innovation. At the Institute for RNA Innovation, we study all the ways that RNA can be used as a therapy. The institute itself is massive. It spans campus. The physical institute is in one spot, but the number of scientists that are coming together to do this work come from all walks of life. We've got people who are basic scientists who understand how does a cell work, all the way through to clinician investigators that are trying to really understand how do we move these therapies into humans, or how do we test these vaccines in order to help people. I'm an infectious disease doctor, so when I take care of patients, I see people who have various infections. And the virus that I studied, the majority of my career is HIV. So I use the RNA platform in order to study, prevent, and treat HIV. So how can we make that next generation of HIV vaccines, that next generation of HIV therapies? We're doing the first studies to see, is this even feasible as a new mRNA-based vaccine? Or is this even feasible as a way of treating HIV?
Can we see what that process looks like in the lab?
Absolutely, I would love to show you. The process of making an RNA vaccine, for instance, actually just begins at the computer, where we decide what is the vaccine that we're trying to make, for instance. And once we do that, we make a piece of DNA called a plasmid. That plasmid then serves as the template for RNA, meaning that DNA is used to make whatever RNA. And so we have many ways that we can make RNA. A lot of us make RNA by hand. And so this is one of our clean rooms where we make RNA. And you can see these various shakers, and you can see the centrifuges that we use. This is the essential equipment that allow us to make RNA that's incredibly high quality. But I want to show you the future, what we're really excited about. We have been working to make a robot that will be capable of making many RNAs at once. And so here is the RNA robot. This will hopefully allow us to make tens or even hundreds of RNAs in a single day. And this will allow us to test more and more different types of vaccines and mRNA therapies. So once an RNA is made, we have to purify it, and that we often do by hand. And once it's ready, we have to test that it makes the protein that we want it to make. And so the question is what is the best way of testing that? So often what we do is we take cells and we put the RNA into those cells. And so the place that we do this is in the tissue culture hood. So I'm just gonna open the door here so you can see what tissue culture looks like. So these large hoods allow us to keep everything sterile. And down at the end, we have our incubators where our cells live. And so once we transfect or introduce that RNA into the cells, we have to wait, because the cells are going to start making the protein. And once they've made enough protein, we have to evaluate that protein to see if the correct protein of the correct size was made. There are many ways to ask, was a protein made? Was it the correct protein? So one of them is with a Western blot. But one of the really cool other ways that we can do this is with something called a flow cytometer. And so what a flow cytometer does is it allows us to look what proteins are on the surface of the cell or inside of a cell. So when we have a cell that's making our vaccine, we stain that cell with antibodies, and then we ask, is that protein present? And so this is our flow cytometer. It's an incredibly fancy machine. And you can even see with this platform here, we're able to sample 96 samples at once. And the machine just moves from well to well. Within the span of about 30 minutes, we could evaluate 96 different vaccines using this technology. Once we're done ensuring that the RNA is made properly, we have to put it in lipid nanoparticles. Lipid nanoparticles are really good at making vaccines. They give a really good immune response, but they also protect the RNA because there's a lot of things in the environment that break down RNA. And so when we formulate in lipid nanoparticles, which are these tiny fat droplets that are super small, we have to use microfluidics, which are these tubes that take the lipids and the RNA and push them together at the right speed. So here is the machine where we make lipid nanoparticles. And once we have a lipid nanoparticle, then we go test it. So maybe we'll go into a mouse and see, does that make an immune response in the mouse?
I had a great time touring the Penn Institute for mRNA Innovation, and I hope you enjoyed the tour too. You may be wondering what happens if animal studies, like Ted mentioned, are successful, and that's where clinical trials in people come in. If a potential vaccine seems like it might work after many studies in animals, it will be tested in clinical trials with people. Clinical trials occur in several phases, with increasing numbers of people each time, and they often take several years to complete. To find out more about clinical trials, check out the Vaccine Education Center's webpage at vaccine.chop.edu/vaccine development. Thanks for taking the time to learn about studies of mRNA technology.